Good afternoon and welcome to today's public talk from the NIHR Oxford Biomedical Research Centre. Today we're hearing from Professor Andrew Pollard, who is Professor of Paediatric Infection and Immunity at the Oxford Vaccine Group. Andrew is also co-theme lead for vaccines at the Oxford Biomedical Research Centre, and he is currently jointly leading the Oxford's COVID-19 vaccine trial with a number of colleagues. Today he'll be speaking to us about the vaccine trial and the importance of developing multiple vaccine candidates for COVID-19. If you have any questions for Andrew, you can submit them over the YouTube live chat, or you can email them to us at obrcinquiries@ouh.nhs.uk, and that email address is also in the video description. I'll hand over to you now, Andrew.
Thank you very much, Kelly. So I'm going to talk today about coronavirus vaccines and the huge efforts that are going on around the world to try to develop vaccines that provide us a way out of the pandemic. And I think everyone will be aware that there have been an enormous number of cases around the world. Here you can see on this map from the World Health Organization that there are about 12 million documented cases and well over 500,000 deaths. But of course, this is just reflecting where testing has been done to demonstrate those cases have occurred. And there are many cases that remain undocumented, particularly in parts of the world where there's more limited access to testing. And as everyone here in the UK knows, and it's true around the world, at the start of the pandemic, there was relatively limited availability of testing everywhere. So many cases are unrecognized. I show on this slide from Johns Hopkins University the countries at this moment that are the worst affected by coronavirus infection. And you can see here this big increase in cases in the United States and in Brazil. And the particularly worrying aspect here is also the big increase in India and, as you can see, in South Africa, showing a sustained increase in cases that's been occurring over the last month, which really suggests that attempts to contain the virus continue to be thwarted in many parts of the world. And of course, this is because this is a virus which is very successful in transmitting from person to person. And although in the UK, where we've recently come out of lockdown, which was very successful in controlling virus transmission, and it feels like it's over, the reality is the virus is still out there, and relatively few people in most populations have been infected so far, which means that the pandemic has still got plenty of time to run. Another really important aspect of the pandemic, which we're all increasingly aware of, is this difference in the risk of disease dependent on age. And you can see here that if you look at the infection fatality ratios, that the risk is very, very low amongst children of severe outcomes. But in those who are older adults, the risk is extremely high. And my teenage son was delighted to tell me that my chance of dying if I develop coronavirus is about 100 times more than his. Of course, the thing that we all have wanted to know as the pandemic hit is what's going to happen next. And it is extremely difficult to predict the future, as we've seen. We didn't know quite how successful lockdown would be in the countries that have been able to implement it. And similarly, it's very difficult now that many places are emerging from lockdown and what the future will hold. And here, in a report just published today from the Academy of Medical Sciences, are some predictions of a worst case scenario. And this is where the R number, which you can see on the left here, is at 1.7. So at the moment in the UK, we're under 1 in our R number. But if it were to rise, now that we've come out of lockdown, to 1.7, then this is the sort of scenario which could be predicted, with infection rates rising to levels perhaps not as high as the peak we saw earlier this year, but a much broader peak, so that over that period of time there would be many cases. And similarly, on the top right panel, also a broad number of deaths in that period. And of course, when you look at beds occupied, this both in hospital and in critical care settings, this would have an enormous burden on the health system. Now that at 1.7 seems quite a long way from where we are at the moment, which is well below 1. But remember that at the peak of transmission earlier in the year, we were nearer 3 as the R number, the transmission rate. So this is what's considered to be perhaps a worst case scenario where there's some control of transmission, but it's not perfect. And the various mitigation measures that are in place, including social distancing and so on, would have an effect on it. This is perhaps a more modest scenario where the R number is 1.5, and you can see here that the infections would be much lower than were seen on that previous slide, but there would still be a prolonged period of time where there were many more cases than we're experiencing at the moment in July of 2020. So these different scenarios are predictions of what might happen in the months ahead, and the extent to which local lockdowns and isolation of cases and management of the pandemic virus over that period will impact on this has been worked on to try to make some sense with these models. But they are models that try to predict what will happen in the future. And of course, it's very difficult because it's so dependent on the way the virus behaves, but also how the population behaves and the sanctions that are put in place to try to control the virus. Another really important aspect just to raise briefly, and I won't go through this slide in detail, but the point that it's making is that in developed countries, particularly in Northern Europe and North America, there is a relative social isolation of older adults compared with younger adults. And we've seen this recently with partying younger adults, with older adults much more nervous about venturing outside. And of course, this is normal in normal society in northern hemispheres. But if you look in most developing countries, that mixing pattern is rather different. And the families tend to live together with all generations in the same household. And this is, during the pandemic, also reflected in transmission, in that although older adults in our society have much more severe disease, they are relatively protected from disease by being more socially isolated normally. But that is not the case in the developing country setting, where there is equal transmission regardless of age. So we do have the possibility of treatments which might make any of further waves of disease less severe. But perhaps the best way to be able to find a route out of the pandemic is through vaccination, by generating the immunity that could protect against the virus. And different strategies might be used. We might vaccinate whole populations to try to completely end the pandemic as quickly as possible. And that has the advantage, as if you vaccinate enough people, you can completely stop transmission of the virus and allow those who cannot be vaccinated but remain susceptible to be protected, because the virus just can't ever get to them because it can't transmit through the healthy population. So that's an ideal scenario where a vaccine is perfectly effective. But it may be that, at least initially, with some vaccines there's constrained supply, and it may be more logical to target those who are in risk groups, either because they're at risk of disease, like key workers who are in the front line, whether they're working in a hospital or in other settings where they're more exposed to people who could be carrying the virus. And then also those who are at risk of severe disease, such as older adults, and those who have other medical conditions which put them at greater risk, or perhaps other risk factors as that there are, that perhaps even some that we're not aware of yet. So where are we with vaccine development? I just checked in this morning with the World Health Organization, and there are now 23 vaccines that are already in clinical development. That means in trials involving people somewhere in the world. And behind that are another 140 vaccine candidates that are being prepared for clinical development. So this is an absolutely unprecedented situation where, in a matter of 5 or 6 months since the start gun, there are already 23 vaccines that have got into humans that are being tested to see whether we can protect the population. And so that, I think, should give us a lot of optimism that perhaps one of these vaccines, or maybe even multiple products, will have a hit on target and be able to help in controlling the pandemic and move us forward into a world where we can get back to normal. Now, just to remind you of the virus, this is what the virus looks like, and you can see it's decorated on the surface by these red blobs, which is spike protein. And the spike protein is critical to the ability of the virus to transmit, because it's the spike protein that binds onto receptors on our cells when it gets into our airway and allows it to get into those cells and cause an infection, and particularly to get into the lungs and cause the severe disease and the pneumonia that results in people being admitted to hospital. And we knew from January what gene the virus had that made this spike protein, and that gave us an enormous head start, not just us, but all vaccine developers around the world, to be able to very rapidly come up with vaccines to try to protect against the pandemic virus. The other thing that gave us a really big head start was the fact that this is not the first epidemic of a coronavirus. We've had a number in recent years, and I'm just highlighting the two that are the most prominent. 18 years ago, SARS was the first coronavirus to cause severe cases and deaths, about 11% mortality, so much more dangerous than the current virus. But fortunately, it didn't spread quite as well as this virus, and it was possible to contain it using isolation of the cases. But this perhaps was the first warning, and work started on coronavirus vaccines driven by this worrying experience with the SARS coronavirus. And we learned a lot about the biology and what might be needed to protect against coronaviruses from this outbreak. The next really important one was MERS coronavirus, which almost certainly jumped from camels from about 2012. And this virus had a very high mortality. Over a third of people who were infected died, and created enormous worry around the world that coronaviruses might be important in causing severe disease and spread. And the MERS coronavirus outbreak also drove huge efforts in vaccine development. And my colleague here in Oxford, Sarah Gilbert, was working on a MERS coronavirus vaccine that really gave us an enormous leg up in being able to switch to working on this new pandemic virus. And here you can see on this slide a whole series of different viruses and bacteria which cause outbreaks. And since 2015, with government funding, investigators in the UK have been working on trying to make vaccines against all of these different diseases because of the concerns about global or local outbreaks that could threaten human health. And as I mentioned, the MERS coronavirus was already being worked on here in Oxford by Professor Sarah Gilbert, but also the vast majority of the other bacterium viruses on this list were being worked on by myself and colleagues around the university, and particularly here in the Oxford Vaccine Group and the Jenner Institute. Now, there are a number of different types of vaccine which are being developed, and I'm just going to very briefly touch on the different types that there are. The first is our inactivated virus vaccines, and in this situation the virus is taken, the coronavirus, and grown in the laboratory, and then it's chemically treated to inactivate it so that it's a harmless product which can be used as a vaccine. And this is very similar to the traditional types of vaccines like the killed poliovirus vaccine, which we give to all children in the UK at the moment. So this is a very standard method of making vaccines, and quite a number on that list of 23 which are in clinical development take this approach. Second are protein subunit vaccines, and here the spike protein from the surface of the coronavirus is used to make a vaccine. And as I mentioned before, the spike protein it seems to be very important for the virus and its transmission. And when we make immune responses against the spike protein, those immune responses are able to neutralize the virus and to stop that virus from spreading and getting into cells. So the protein subunit vaccines, again, a very traditional technology used in a number of different types of vaccines already that has been developed, and a long list of developers are working on this approach. Next are the RNA vaccines, and the genetic code of the coronavirus is based on RNA, which is a type of genetic chemical that's used to give the instructions to the virus about how it should divide and make new viruses when it gets into cells. By taking the RNA from the virus, it's possible to make a harmless vaccine that cannot cause infection, but the RNA itself, when it's injected, gets inside our cells and then makes the spike protein, because it's an important messenger that is used by cells in order to then form into proteins, including the spike protein. So the RNA vaccines, and in fact an RNA vaccine was the first coronavirus vaccine to go into trials in Seattle in March. So these vaccines, there are a number of different types of them, and quite an exciting technology that has potential not just for coronavirus but for future vaccines that may be useful for protection against other outbreaks. The next type of vaccines are DNA vaccines, and this is another type of genetic material. Again, these vaccines are designed for the DNA to get inside our cells, and that DNA then gets converted by our cells into RNA, and that in turn gets converted into the spike protein, again so that our immune systems can recognize the spike protein to make an immune response and in the future protect us from coronavirus infection. And lastly on this slide is the type of vaccine that we've been developing here in Oxford. And in this case, the gene that makes spike protein from coronavirus is taken, and you can see this gene on the left of the slide here and the spike protein underneath. And this gene is inserted into a type of common cold virus called an adenovirus. And after the gene has been inserted in there, that common cold virus is then made into a vaccine. Now that virus has been deactivated so that it cannot cause infection in humans. And so if this vaccine is used to vaccinate individuals, the virus gets taken up inside cells and leads to the expression of spike protein so that we make an immune response against coronavirus, but is completely harmless. It can't cause any ongoing infection of the individual who's vaccinated. So these viral vector vaccines, like the one being developed here in Oxford, are a great way of shuttling a new gene to make a protein into our cells so that we can make an immune response, and in this case against the coronavirus. Now the problem really from having got the design of a vaccine is the normal development cycle, which normally takes somewhere between 5 and 10 years from the vaccine design stage through to having a licensed vaccine that can be used on a large scale in the population. And clearly in a pandemic, if we want to have any chance of ending it and getting back to normal life, we can't wait for 10 years. And so we have to unpick what many of the roadblocks are that mean that it takes so long in order to be able to make the coronavirus vaccine. And so rather than going through this very lengthy program from design through the different phases of clinical trials, through to eventually, at the end of all of that, starting to make vaccine at scale, a lot of these different stages have been happening rather in parallel. And so the vaccine design, as I said, was we had a shortcut because we already knew how to make the vaccines. And whilst the first manufacturing was going on to make vaccines ready for the first trials, the animal studies were being undertaken for the vaccine. And on the 23rd of April, using the data that we had from animal studies plus this new manufactured material, we were able to start clinical trials here in Oxford. That process up to this is very carefully regulated, and the way in which it could be sped up wasn't because of cutting corners. It was because the regulator was able to put all efforts to make sure there are enough individuals to review all of the data and make sure that the quality of the product was exactly as it should be. And our first trial, which was a phase 1 study, we expanded to 1,000 individuals between 18 and 55 years. And the main questions there from this study was, is the vaccine safe, and do we find an immune response? And we're hopefully going to be able to release data about the phase 1 trial in the very near future, because we've done some analysis of the early data that we have, both on the safety and the immune response to the vaccine. But of course, this part of the study is in younger adults, those between 18 and 55. But a really important question, given my comments earlier about the severe disease in older adults, is to look in other age groups. And we have another study called a phase 2 study, where we're examining the immune response in those over 55 and also in a group who are over 70 years of age. Because it is known that with most vaccines, immune responses are much poorer in older adults than they are in younger adults. And so we need to understand that and work out how best to optimize the magnitude of the response that we can make to the vaccine, with the aim of being able to protect people. And then lastly, moving on to the phase 3 studies, these are undertaken in much larger groups of individuals to assess whether or not the vaccine is safe still, but also critically whether we can protect against infection. And so in all of these trials, we divide our volunteers into those who get the new vaccine and the other half who get a control vaccine that does not protect against coronavirus. And by having those two groups, we can count the number of cases that happen in each arm of the study and make an estimate statistically of how effective the vaccine is. And of course that takes time, and we're now just accumulating data in the phase 3 trials, which began at the end of May. And we will have around about 10,000 people vaccinated here in the UK, and we're currently expanding trials in other countries as well. Now, while these clinical trials have been going on, the manufacturing has also been going on, so that if we are able to show protection in the clinical trials, we'll already have vaccine made. Clearly, that's a very expensive process, and if the vaccine doesn't work, then there will be a lot of money having been spent on making the vaccine. However, I think everyone recognises that in this situation, it is a good idea to continue manufacturing at risk, because if we do have a protective vaccine, it has enormous potential for helping populations escape from the pandemic. So I mentioned that there are other trials which are underway internationally. We're working with colleagues in Brazil and Sao Paulo and Rio on a trial of the vaccine in that setting, and we're currently enrolling there and aiming to enroll about 5,000 people over the course of the next month. And in parallel, we have, working with colleagues in South Africa, have started enrolling 2,000 people, and we hope to complete that enrolment during this month as well. And so with this, we'll be able to assess how well the vaccine works in different populations around the world, and also here in the UK in a large population here. We have also initiated a partnership with the university has with AstraZeneca, and they will next month start trials in the United States, so that we'll have a lot of data from different populations about how well the vaccine performs. So what of the future? An effective vaccine could really make a big difference if it was able to prevent a second wave of disease. It could also be used for control in high-risk groups, those who are at particular risk of disease, as well as those who have remained shielded or socially isolated to protect them, allowing them out from that if we could find a protective vaccine. There really is a huge effort still needed to get the answers that we're all waiting for about how well these vaccines work, how long does protection last, and so on. And those results are absolutely critical to be able to move forwards and to protect populations. And we also have the very good news that there are many other vaccines that are being developed in parallel. And because many of them are focusing on spike protein and will be making very similar immune responses, if we do get a hit on target, it's very likely that that will translate into multiple hits on target with the other vaccines and allow us to be able to deploy vaccines around the world much more quickly than if we only had solitary efforts going on. As I said, these efforts only started a few months ago, and it's incredible already to be in a position where we should have the first data available from these vaccines very soon. And I'm going to end there, and thank you all for listening.
Thank you very much, Andrew. That was extremely informative, and it's clear that there's so much hard work going on in this, so thank you very much for taking the time today. We've had some questions, so I'll get straight into them. First of all, I think it's the question on everyone's lips is when we can expect a vaccine. Has that changed over the last few months in terms of what you're expecting?
Well, you're asking the most difficult question possible because it's all about predicting the future. As you saw in the first few slides, there's a lot of uncertainty still about what happens next. If there are lots of cases over the months ahead, it'll be relatively easy to tell whether the vaccine can prevent those cases. But if we have a very low level of disease, it will take longer. I mean, we do think that this virus will continue to spread in human populations. So over time, we will have an answer. But the speed at which that happens depends so much on how the virus spreads, what people do, what happens post-lockdown from a UK perspective. And similarly, the transmission in other countries where we're working, we know what's happening today in those countries, but we really need to know what's happening in the next 3 or 4 months in order to answer the question.
Okay, thank you. I'll ask you a couple of questions now, they're related. Firstly, how long do you expect immunity from the vaccine to last? And is it possible that the vaccine will generate antibodies that last longer or behave differently to those generated by the actual virus?
Well, it's a really important question. I mean, we don't know how long any protection will last, and we don't know what level of antibodies are needed in humans for protection. I think the one thing we can say is that with similar types of vaccines, immune responses seem to be quite long-lasting, but that doesn't necessarily mean that they'll be protective. And of course, one of the great things with vaccines is if it looks as if immune responses are waning, and we eventually know what the protective level is, we can give booster doses in the future to try to maintain protection. It is true that there are emerging data suggesting that immunity after infection may start to wane relatively rapidly, and that's been well recognized in the past with coronaviruses, so is perhaps not so surprising. It isn't necessarily the case that vaccines will induce the same sort of immunity as the disease. And so I think I'd be surprised if things are exactly the same, but we need to actually have a bit more time pass to allow us to do the measurements to answer that question properly.
Yes, of course. With all that in mind, do you think that a vaccine will allow us to return to normal, to how things were before, or will there still be a need for things like masks and social distancing, even when we are starting to vaccinate?
Well, I think the answer to that question really depends how effective the vaccines are and how much supply we have in order to protect people. I think that if vaccines are very highly effective and we can prevent severe cases with them, then one would anticipate returning to normal. But a lot of that is caveated around how much vaccine there is, how effective it is, and how quickly we can deploy it. And what recommendations are about who should be vaccinated to really fully answer the question. I think one thing that to me seems likely is that the world is going to be different after this pandemic anyway, but what that actually means in terms of how we behave as human populations, I rather hope we do get back to something a bit more normal, and hopefully that'll be fairly soon.
Yeah, I imagine we are all hoping that. how quickly can we scale up manufacturing for the vaccine? And similarly, how long might it take to vaccinate a whole population, as that was one of the options that you described?
Well, I think upscaling of manufacturing is going on around the world, not just for the Oxford vaccine, but for other vaccines as well. And so I think that we'll be in a position relatively rapidly where there are large numbers of doses available. But you know, this is a big requirement to do that. Most vaccines that are used at the moment in the world are for young children, which is a relatively small number of doses, still very large numbers, but small numbers of doses when you compare that with the 7 billion people on the planet. So it very much depends on what the strategy is for controlling the virus. If we had to have enough doses for everyone in the world available, that's going to take quite a long time. If we're saying that we're going to focus on a subset of the population, at least initially, then that becomes feasible in a much shorter time frame. For the vaccine being developed here in partnership with AstraZeneca, there is work going on to manufacture all over the world at the moment to try to make sure we get there as soon as possible. Thank you.
You've mentioned obviously the other COVID vaccine trials that are happening around the world, including this one at Imperial in London. How are you collaborating actively with these rival trials, or is it more parallel development?
Well, we talk a lot with the other developers and look at what actually we can learn from each other in order to try and move as swiftly as possible. I don't think we're in a competitive environment here. We're all trying to make sure that we can deploy vaccines that can control the pandemic virus. And certainly from the university's perspective, the critical thing here is to be able to protect human health, and that's why the focus of the partnership with AstraZeneca is a not-for-profit partnership.
Thank you. Someone has also asked, how do mutations of COVID-19 affect vaccine development? Can you talk a little bit about that?
Well, it's a really good question. I mean, at the moment, we're not aware of mutations which would stop vaccines from working, but that is something which will have to be watched over time, because there are certainly some viruses, and the best known is influenza, which changes its spots every year, and so it's quite easy for it to escape from vaccine protection over time. With the coronaviruses, so far, we're not aware of huge changes over time, but if that did happen, then it may be necessary to have a strategy where we followed rather like flu, where you have to make a slight tweak each year to have a vaccine that's protective.
Mm-hmm. And we've also been asked, do your team plan to try boosting vaccines, for example, with COVID-19 encoding MVA or other products?
Well, I think that the first answer to that question is that we're already looking at boosting but 2 doses of the same vaccine. And that has some advantages in that it's much easier if you're trying to run a public health program just to have one product and to give 2 doses of it. Because it can be quite confusing at the delivery end of this if you've got multiple different products that have to be given at different times. But certainly there is evidence from other vaccine strategies that when you give the first dose with the type of adenovirus that we're using in our vaccine, and the second dose with another type of viral vector, another virus that can shuttle the coronavirus gene into the human body, like MVA, and then that can give better responses. So that is that sort of thing, and also looking at perhaps doing studies where we give 1 dose of the Oxford vaccine, another dose of the Imperial vaccine. All those things will be extremely interesting to look at over time. But that's going to take a while to put all of that together, to look at what the best strategies are for protection.
Yes, of course, thank you. We've asked, is there a known difference in response to vaccines between men and women or people of different ethnicities? Is that something that you have found?
Well, we haven't looked so far in the current studies, but it's an important question. We certainly know that women tend to make better immune responses to vaccines than men, which I have to say I've always found a bit disappointing. But it's not true for all vaccines. But it is something which we've observed with a wide variety of different vaccines, and will be interesting to observe. Whether that really makes any difference in the level of protection, I'm not sure. These are measurable differences, but probably not important differences. As far as different ethnicities are concerned, that's much more difficult because most of the studies have been confounded by them being in different populations, and we definitely see different immune responses in different countries, but that may be not a result of the ethnicity differences, but more to do with other environmental factors in different countries. And there are very few studies which have attempted to systematically address that question.
Great. And I think I'll just ask you one last question now, as you've answered so many for us. A few people have been asking, you've mentioned about the phase 1 results. Have we got any clearer idea of when they will be available?
Well, I hope it'll be very soon. I can't give you a definite day yet, but we're making progress in that direction.
Great. Well, thank you so much, Andrew. We really appreciate it. And thank you, everyone who's joined us today. Thank you.